Imagine the one you desire is a captivating specimen. Yes, this skin specimen or this onion specimen. Imagine the light microscope. If we see their skin glowing under the light microscope, notice the outlines of cells. In skin cell of an onion, we can see the nucleus, cell wall, cell membrane, and vacuole. Now, let's talk about our belief towards cells under microscope. Instead of saying, I'm not interested, think, I'm mesmerized by the beauty of these cells. Now, imagine the electron microscope. Zooming deeper, discover the contours of their innermost shapes. Deep down, the inside of their cells. Yes, inside of theirs. Under electron microscope is the finer details of the cells, organelles to be exact. Here we can see the mitochondria, endoplasmic reticulum, nuclear pore complex, nucleus, and nuclear envelope. This is possible because electron microscope has greater resolving power and higher resolution. Again, belief. Instead of saying I'm not interested, think I'm curious about what details make cells beautiful. Actual cell size. We can calculate the actual size of a cell by identifying the magnification. The magnification is equal to image size over object size. If we are going to arrange this, we can identify the cell size, which is equal to image size over magnification. Usually, cells are a few micrometers long. 1 micrometer is equal to 1 times 10 raised to negative 6 meter, or is equal to 1 over 1000 millimeter. It means if you use a ruler, you have to divide the 1 millimeter by 1000, and that is the size of a cell. Now, how do you see this onion specimen? Remember, light microscope is the outline of cells, while electron microscope is the organelles of cells.